Alrighty, it is now 2 o'clock, so let's get started. So welcome everybody to the second installment of the EPIC summer seminar series. I'm Jonas, and today we should be streaming from 3 platforms, so we avoid the technical issues that we had the first talk, but the first talk should be uploaded to YouTube, so you can go and watch it there. And this talk will also be uploaded to YouTube afterwards, so you can go and watch it there. Alrighty. So first I will start to talk a little bit about what EPIC is. So EPIC is short for Evolution and Population Genetics in Copenhagen. And we are a small network. It's a joint effort by the Statistical and Population Genetics group in University of Copenhagen and the Globe Institute. And what we do is that we usually would have monthly physical seminars where we would have scientific discussion and socializing, and usually some pizza also. But we cannot do that right now, so instead we have decided to have this summer seminar series. All right, and if you are Copenhagen based and you want to join this network, then we have a mailing list here. You can find the link, which is just pubgen.dk/joinepic. And if you want to read more about all this and find the schedule and everything, you can go to our new website, pubgen.dk/epic, where you can also find all the streaming links. Yes. And this is just the program for the seminar series, so you can see it. So the first talk was Monday by Rasmus Nielsen. Today is Benjamin Peter, and so on. And all this schedule and so on can you also find on the website. All right. And today's talk is going to be by Benjamin Peter, who has also been teaching quite a few times at our PhD summer course. And Ben, he did his PhD in Berkeley under the supervision of Rasmus Nielsen, who was the first speaker Monday, and Montgomery second. And then he also did a postdoc at Chicago with John November. So he's been surrounded by very great people. And now he's a group leader at the Max Planck Institute at Leipzig in Germany. And he has done a lot of great work on selection, population structure, integration, and so on, and developing methods and theory in the field of population genetics. And today he will be talking about gene flow between Neandertals and Denisovans. And with that, I would like to give the word to you, Ben.
Okay, can you hear me?
Yes, we can.
Okay, just excellent. So I'm just gonna see, and you can see my slides hopefully, or my first slide. Anyway, thanks a lot to Jonas and Raz and Hernan and Anders and Ida and Fernand and everyone else that was involved in organizing this. It's a great pleasure, even though I cannot be personally in Copenhagen this year, unfortunately. It's so nice to give this talk, and it's going to be a great lecture series, and I hope I can contribute my part. So I'm want to give a talk about a project that's been going on for a bit over a year now, where I look at gene flow from ancient DNA. There's a particular focus on modern human history. I should just be able to switch my slides. That seems to have worked. So to start with, this is just a sort of schematic overview about what people currently think our genetic history looks like in this class. I mean, all modern humans, but also our closest relatives, Neandertals and Denisovans, from which we, or at least some of us, have some genetic material derived. So the common model, just to bring everybody on the same page, is that humans, Neanderthals, and Denisovans are 3 fairly distinct hominin groups that diverged roughly 600-ish thousand years ago.
Sorry, Ben, to interrupt you 1 second. We have a weird background noise on your side. I'm not sure if it's your headphone set.
A weird background noise. That's very unfortunate.
It only happens when you speak.
Is it better,
So
like...
maybe it's your headphone set, like your microphone.
Okay.
You could switch your microphone settings to be your computer, the microphone, rather than your headset.
Yes, I think that's what I've been doing. I've just unplugged my headphones. I'm not sure. I see. So you have some background noise.
Yeah, some kind of, it sounds like a fan. It's not a fan, but it sounds, every time you speak, sounds something slightly weird.
Okay. That is very unfortunate. I'm not quite sure if I don't think of another computer I can use easily here.
Can you speak now to see if it's still going?
Yes, can you hear me?
I think it's a bit better. Maybe you can go ahead and I can interrupt you again in case it comes back or something.
Yeah, that's not, sorry, I should have tested that a little bit better before. It's weird that that didn't show up before when we chatted. Anyways, yeah, sorry about that. Yes, so exactly. So essentially what I wanted to start with here is a model of our human genetic history, where we think that Neanderthals and Denisovans and modern humans diverged roughly 600,000 years ago. And then somewhere around 400,000 years ago, Neanderthals and Denisovans diverged into two distinct lineages, where Neanderthals are fairly well recognized. First Neanderthals have been recognized well over 100, I think almost 200 years ago now. And there has been tons of fossils, both bones and stone tools associated with them that are well studied, where in contrast, Denisovans are a very new group. They've been mainly recognized using from genetic data. Only using genetics has been shown that they are very distinct from Neanderthals. And the fossil record is very meager in the sense that it's like a bunch of teeth and tiny bone fragments that are firmly associated with Denisovans. And to understand, what makes us humans, we need to understand our history, right? And so there's a tree-like base structure in this graph, but there's also lots of arrows that go between lineages that denote gene flow or contact between some of these groups. And this is sort of one of the main things that I've been interested in the last couple of years, or really since I moved to Leipzig, and how we can quantify and infer that gene flow. And that's really like the reason we can do this inference, I think, is really due to like major advances in the technology of ancient DNA. So DNA retrieved from fossil bones and teeth, right? And this is a field that's still astonishingly young and have made immense strides in the past decade. So for example, if you look at all the ancient DNA human samples that have been published, genome sample has been published in like 10 years ago. If you look at the sample of what happened in was a label in 2010, you just see like one single dot of a Neanderthal gene, the first Neanderthal draft genome that's been published. Then if you look at how this map looked like 6 years ago, so in 2014, which it has built in a little bit, but there's still like maybe a handful of points. And it's only if you go to 4 years or 2 years ago, or I haven't updated the map in some years, but the number of points that one gets grows immensely. There's several thousands of samples that have been published, and most of them are publicly available, and we can use and reanalyze them. Now, this gives us a great opportunity to study our genetic history and learn from direct observations in some cases, which populations were in which places at which times. But it also harbors some major technical and methodological challenges, and very hard work, that need to be overcome. And so maybe the most obvious challenge is that ancient DNA is often very low coverage, right? So coverage of a genome is simply how often each base pair is seen in the genome. So each base, so in a short read sequencing, molecules. So the higher coverage one has, the better quality the data is. But if one looks at the coverage of most of these ancient DNA genomes, so this is a summary data set that's been compiled by David Reich's group that I'm using here just to give a broad idea. If you look at the coverage here on the x-axis versus the frequency of samples on the y-axis, one sees that the vast majority of samples have a coverage of around 1x or a handful of x. And while there's a few high coverage genomes, which high coverage typically is everything above coverage of 25 or something around that regions, the median coverage of ancient genomes is, in this data set, is I think 0.07. So less than 1x, meaning that most of the, or a lot of the DNA will be missing, and you have to deal with that. And me and many others have been working on tools and methods that can deal with that to still make use this, in some sense, crappy or sparse data for inference. And that's what I'm going to talk about today. And the application I'm going to talk about is exactly this admixture. The signatures of this admixture, these arrows or this gene flow between different lineages. And if you think about, or if you look at on a genomic level, what the result of this admixture will be chunks of genomes in every single individual, or a descendant of one of these admixture events that will come from a distinct group. So, for example, here I just showed an example from a paper by Bernard Akey, published in 2014, where I looked at Neanderthal ancestry in present-day Asians and Europeans. And you can see here 22 chromosomes, and in blue and in red are all those genomic regions where they identify some Neanderthal ancestry in these populations. And we can see that there's quite a lot. In some region there is none, and this is then used for follow-ups in terms of functional analysis, or also to learn about where and when this gene flow might have happened. Now, this is all very nice, and there has been at least a dozen methods that do that, but it becomes quite hard if you want to run these on ancient DNA. And so this makes it also quite difficult to do this type of inference about admixture in ancient genomes. And just to maybe briefly show how these patterns originate, this is the toy model that one can think of in the context of this topic of the Denisovans and Neanderthals. But for the previous level of the Neanderthals and modern humans. So what happens is that Neanderthal and Denisovan will have an offspring, right? And so in their genomes, they just have each line here corresponds to a chromosome, and everything in different organisms as each chromosome has 2 copies. And so what will happen is that the offspring of a Denisovan and Neanderthal will have 1 chromosome from its, say, Neanderthal father and 1 chromosome from its Denisovan mother, and it will have these sort of mixed carrier type for just for 1 chromosome, but obviously the same is true for all the other chromosomes. And then if this individual, say, interacts with other Neanderthals in this case, then recombination will split this up into smaller and smaller. Right, so in this case here, after 1 or multiple generations, this blue and yellow one individual will create a gamete or an offspring chromosome that is missiled at 1 to 1. And if we wait for more and more time, for tens or hundreds or thousands of generations, these fragments will become shorter and shorter, and this will further or more and more across the genome. And so the point really is that we can use this fragments to learn about when and where genes will happen. The issue with ancient DNA that I'm going to talk about here is really that coverage is the main issue, that often we don't have often only don't have complete genotype information. Another issue that is very prevalent is contamination. So very often these ancient DNA data, right, comes from bones that have been in a cave or in the sediment for tens of thousands of years, which is not ideal for DNA storage. And so some changes will happen, and people have handled them, and so one needs to take that into account. There will be often some ascertainment bias, because to combat this contamination and low coverage, people often try to enrich for very specific parts of the genome, which allows us to get much higher coverage for specific samples, but at the cost that we only get specific sizes that have been chosen by the researchers. And so as a result, one thing that one often cannot do with ancient DNA is to phase the data, right? So even though schematically we think about there's two chromosomes that are homologous to each other, that have the same genetic information, in practice we can usually not figure out for each sequence read which part of which chromosome it comes from, which is something you usually can do with fairly high reliability for present day data. And so as a result, the model I'm using sort of has to incorporate all these issues you have in ancient DNA. And so without further ado, this is the basic schematic of the model I want to talk about. It's a HMM or graphic model using HMM that has two layers or three layers. At the bottom layer, you have local ancestry. So this is what you're interested in. This is this coloring or painting of the genome into where any particular part of the genome has some Neanderthal ancestry or Denisovan ancestry or possible more human ancestry. And given we know the local ancestry, we can imagine ask what's this individual's genotype at this particular locus. And the way we do that informally is to just look at other Neanderthals or Denisovans and ask, is it similar to those, and derive a likelihood for that. And then based on the genotype and what we think we know about contamination, we can ask then what types of sequencing reads or sequence molecules would one expect from this individual, right? Taking into account that it will be damaged and fragmented and possibly contaminated. So in a little bit more detail, this is how the model looks like. And we take this entire genome, right? It's hundreds of millions of base pairs long, each chromosome, and subset it into small bits. Most applications here will be 0.002 centimorgans, which is roughly 2,000 base pairs. So it varies quite a bit according to every combination map. And doing that allows us to use models for a homogeneous and Markov model, which is just simple, easy for estimation. And the nice, sort of what we gain from that is that we can estimate essentially all parameters pertaining to drift or contamination and admixture timing and so on directly from the data. So we don't need to make strong assumptions that admixture happened at any particular time in the past, as many previous approaches had to do. We can all learn this all directly in the data of this one run of the program. And as I said, so at the bottom here, I have a sort of schematic of the ancestry, right? You have some parts where then you have Denisovan, two Denisovan copies, and then the second bin you would have a Neanderthal and Denisovan. I hope you can see my mouse pointer. And then at some point you have homozygous Neanderthal copy or homozygous Neanderthal region, and so on. And as I hinted at before, we simply model that as either homozygous region or heterozygous region, which I very often will abbreviate as 3-letter codes then for Denisovan ancestry, NIA for Neanderthal ancestry, and concatenation NIA then for heterozygous ancestry. And of course, we should also be hopefully mostly consistent to the right. Then we need to, using this model, so if we assume we knew what the ancestry is in a particular window, say for the person we knew that this particular individual we were looking at as Denisovan ancestry, right? And we would have to ask what's the probability of any particular genotype at the position of interest based on the source of reference. So if you have Denisovan ancestry, you only use the Denisovan source. If you think it's heterozygous for Neanderthal and Denisovan ancestry, you would need to consider both a Neanderthal and a Denisovan reference. So the way I did that to keep the model simple is to use a Baldwin-Nichols model, which essentially makes everything based on beta distributions and is a very flexible approximation that one can use to model allele frequencies and their uncertainties. So we can take into account that there is some uncertainty in the sources of Neandertals and Denisovans because we only have very small reference panels for those. We can take into account that there might be some substructure in those sources, and some time might have passed since mixture. So very briefly, this is the only really massive part of the entire presentation. The way we calculate this probability or this likelihood depends on whether the ancestry is homozygous or heterozygous. The one has slight differences. The homozygous case is actually slightly more complicated. Right? So the model we think about any particular site that we look at is that we have a potential source population. Because homozygous for an ancestry, there will only be one source population. That we then have a Neanderthal population where we have some reference alleles in yellow and some derived alleles that we observe in red, right? And so we know that based on this sample, we know that there has been some this SNP or this site we're looking at has been at a particular frequency, and we try to estimate how long, how much this frequency might have changed from the time when that mixture happened. Right? And so there's a parameter here that we call tau that estimates that. And then the second thing in the target individual, the individual we want to look at, we assume it's different for most of the genome. Right? The haploid case is actually also doable and actually easier. Right? And there's essentially two possibilities. Either these two copies we have in this sample are identical by descent, meaning they coalesce or have a common ancestor before the gene flow event, or they don't. That's the other alternative. And if one plugs that in and does some, into this Baldwin-Nichols model, one gets some fairly simple, at least not super complicated closed form solutions for the probability that one has a heterozygous genotype, so G equals 1, given the A and D are the number of ancestral derived alleles in the source population, so the yellow and red dots. F and tau are two drift parameters where F models how likely it is that one coalesces here in the target population, and tau is a measure on how much the allele frequency changes from the source to where gene flow happens. So how accurate in some sense our source population is. And so for the case where it's heterozygous or homozygous, one gets these fairly simple equations that one can easily use, and one can, what I do then in practice is optimize for tau and F while running the algorithm. Now in the heterozygous case, this becomes actually a lot simpler because it turns out that it doesn't really matter, it all cancels out, and I think the logic is quite quite obvious if you think about it, right? So we have a sample from the source population and ask what's the expected frequency or the expected chance we have, say, derived allele at any time in the past, then this will be just a constant because we don't expect allele frequency to change over time. And likewise, because if we have a heterozygous ancestry, we know there's only one haplotype each from each source population, right? So we'll have a second source population. Then we know there cannot be any coalescence event before gene flow, otherwise we wouldn't have a heterozygous ancestry. And so one gets these much simpler equations here that do not depend on the tau and F parameters, which makes optimization quite a bit easier. And so the last bit then of the model, the last piece of the puzzle is the genotype likelihood, where we calculate the probability of the specific reads or bases in a particular read, given the genotype probability, right? So whether it's homozygous, heterozygous, or homozygous for the other allele. And so one thing that I think is quite important here is that we can use non-covariates for contamination, which is the main concern here in genotype likelihood, which is quite different to other genotype likelihood models that people have worked with, where sequencing error, for example, is a much bigger issue, which is something that we largely ignore because we assume mostly ascertained data. And so what we do is we use known covariates like a terminal gemination of reads. So when it has been shown that ancient DNA often has very distinct damage patterns of C to T or apparent C to T substitutions at the end of molecules. And so we can use that then to, for example, group all the reads that we have into reads with a gemination and reads without a gemination and get differences or different contamination estimates, right? So every difference that, say, these reads should have in terms of relatedness to local ancestry should be very helpful in estimating the contamination as well. And we can do the same thing with different libraries that have been, for example, being produced using different prep methods. There are some lab techniques, for example, treatment with bleach or phosphate that can enrich for endogenous DNA. And so like that, you can take these covariates into account by simply binning reads into distinct groups. So that's the basic setup of the model. I hope this was clear. If you have any questions, I will link to the preprint later on. I'm happy to answer your questions. Anyways, so what we get from this, doing all this math and running this program, is something like this. This is a sort of my test individual that I've used quite a lot for benchmarking. It's a 45,000-year-old modern human. It's a high coverage genome. And the way I'm going to present the results, or most of the results, is in terms of these karyotype plots, where I have each bar is a chromosome from the human karyotype, and gray here is just homozygous human or modern human ancestry, and in blue and orange you would have Neanderthal and Denisovan ancestry, and lighter color will be heterozygous regions, and dark colors will be homozygous, or in the case of the X chromosome of this male individual, it will be just the only haploid hole that's there, right? And so what you can see is that this individual has, quite a few Neanderthal ancestry fragments, denoted in these various blue regions. One can see all over the genome, but essentially no Denisovan ancestry. And so what we can do then is for benchmarking, we can compare with other approaches. So here I use a program by Howard Scoff that uses a very different approach to infer archaic ancestry, and but that assumes again that one has high coverage data, which in this case we have. And you can see that the regions were infer tracked. So this is just chromosome 1 to keep visualization simple, but the tracks are largely consistent between two methods. But there's some small differences for very short tracks. We can then ask what happens if you only take 1% of the track, because of the roughly 40x genome that we have for Ustishem, we just take a random subset of 1% of the data. And we find that using this admin program, we actually get very consistent results. Right? So most of the big regions that are present in this genome are still inferred. And it's only if you go down to 0.1x or even 0.01x that this will start to deteriorate. And so at some point, if you have very little data, then one will get less decision. But it's encouraging to me that even with very little data, one can still get a rough estimate whether there has been some mixture, which is obviously really helpful, because particularly for very old samples, it's often extremely hard to get high amounts of DNA. Right, and this also doesn't really depend that much on the ascertainment set. So here I just use 4 different SNP panels that are used. American 3.7 million, 1240K. If you know what they are, then it's good. Otherwise, it's just like different ascertainments. And one gets a little bit of noise depending on how much ascertainment one uses, but the results again are mostly consistent. We can also run it with simulations. In simulations, you have the measure advantage, right? We know exactly which parts of the genome are introgressed and which ones are not. So we can exactly for each fragment of the genome, we can ask what's the is it has indeed correct inferred, is it a false negative, which I would call here in red, a false positive in yellow, and a true positive in light blue. And then there's 3 categories of regions that are classified as introgressed, but not quite right. So one is in pink is a gap. So if one has one long fragment, that's actually inferred as two fragments. Merged is the opposite. If one has two very close by fragments that are actually not overlapping, that are joined together by the model erroneously. And overlap is if one has two overlapping fragments that are joined by the model again. And so this is one of the sort of assumptions or one of the issues with the modeling approach, right? That we don't do phasing, is we cannot hope to infer this overlap or distinguish these overlapping fragments from true positives. And so what we can do here, right, is then look at various samples. So this is a scenario of a Denisovan-like population where integration happened 120,000 years ago, and look at different coverages and samples at different times. Right? And so, for example, if we look at coverage 2, one sees that for very old samples, the Denisovan population here is modeled to be quite a very small population size. One has a lot of genetic drift in the sample are 50,000 years old, so the ones that are the oldest or the youngest, and had like 7,000 years after integration, because most variation from the integration time will be fixed at this time. But one sees that this, as soon as one gets to like 50,000 or very close to the intervention point, that most fragments are inferred very accurately, and particularly fragments that are longer than, say, roughly 0.2 megabase are inferred with this very high certainty. And this is mainly consistent for lower coverage data, even though for very low coverage samples that are very interesting and very long ago, one has very little power as one might predict. You can also look at precision and sensitivity. So precision simply is how many of the fragments that one infers are actually true fragments, and sensitivity is how many of the fragments that there does one actually pick up. And in here I also mentioned the sort of strict positiveness, so how many of the actually called positive fragments are actually true positives, and how many are either gaps or merge or overlaps for the various scenarios. And again, we can see that the model performs not very well for very short fragments. So for fragments between, say, 0 and 200 kb lengths, the model in some scenarios performs quite poorly. But for even 2x coverage data, one gets very high power, relatively quickly for longer fragments. We can do the same thing by introducing contamination, by just adding some reads or fake reads look modern humans. And so here I fixed the coverage at 2%. And it appears that contamination, at least in this scenario, mainly results in lower precision. Higher colors that have higher contamination, that half the data is contaminant. We will have some intermediate length fragment between 0.2 and say 0.5 centimorgans, or 0.2 and 0.5 megabase that might be false positives, but for at least very long fragments, again, this false positive rate is very close to 0. And we also can estimate the contamination accurately. So here I just made 5 different scenarios from left to right, starting from no contamination in the leftmost panel to 50% contamination in the rightmost panel. So each of the scenarios is made up of 10 libraries at 0.2x coverage each, and try to estimate the contamination in each of these libraries in the joint estimation framework. And so gray line is the true, what was simulated, and black is what is estimated, particularly for the higher contamination levels. There is this slight underestimate that I observed, which I'm not 100% sure why it exists. But in general, both the sort of schematic or the general trend between libraries and the magnitude of contamination is fairly accurately inferred. Anyway, so coming after this very long sort of introduction model comes to the meat of the application, what we learned from using this model on data. We're going to mainly focus on the Altai region and Denisova Cave and Biosphère Cave. So to give you a very brief overview about the data that has been retrieved from the Denisovans and Eastern Neanderthals. So this is more or less the set of individuals for which we have full genome data. So in orange here we have Denisovans. In blue we have Neanderthals, and so the nomenclature is sometimes confusing. But Denisova 5 is a Neanderthal from Denisova Cave. Because Denisova Cave is called Denisova 5, and sometimes it's also called the Altai Neanderthal. But here I'll just stick with the archaeological nomenclature. And then Denisova 11 is an F1 offspring between a Neanderthal and the Denisovan. And so for most of these samples, we have genetic data of very varying quality, right? Ranging from high coverage genomes for a few of those, this 30 to 40x genomes, to very crappy data. For example, for Denisova 2, we have only about 0.3x coverage and a contamination rate of about 90%. And so to see then what Talmix produces on this data, I'm going to start with Denisova 11, right, which is this F1 offspring. So we would expect it to be almost everywhere heterozygous. And this is indeed what we see here. So its karyotype looks like this, in that almost everything is in this brown here heterozygous state. But we do find some regions that are homozygous congenital ancestry, which is consistent with the analysis that's been done on this data by Roland when they published this data a couple of years ago. We can then go back to the, and so here just want to briefly go through what happened in time. There's essentially 3 acts to the gene between Denisovans and Neanderthals. And so Act 1 is the oldest Denisovans, Denisova 2 and Denisova 8, both of which are, at least based on the DNA, somewhat simple or similar to each other. And if we look at their local ancestry, we do find that there is, fairly large chunks of Neanderthal ancestry or heterozygous ancestry in those genomes. This is true both for Denisova 2 and also for Denisova 8. Now, the issue is that given that these samples are fairly poor quality, and so this is the contamination estimates we get for example, for Denisova 2. In that on the x-axis, I have split the region where they have this one of these contaminations, which is a signature for ancient DNA or its own in ODM. And on the y-axis is the coverage that we have, and blue and red is the proportion of samples or proportion of reads I infer to be coming from this specimen versus from a contaminant. We feel about up to or almost 90% of all the reads come from not from this specimen. And so one can ask, given this data, how trustworthy are these results? And for that, I do a simple validation analysis on the SNPs in this genome, because I think it's always good, even if a model like Talmix is helpful in detecting regions. It's very important to sort of validate and make sure that what text makes sense. And for that, look at some set of SNPs in the region that have a very specific criteria. Particularly, what I want is SNPs where Africans differ from either Neanderthals or Denisovans, but not both, right? So for here, I simply plot all the SNPs in this region where the Africans have so all Africans that we have in a reference panel have a 0 state, and then either Neanderthals or Denisovans have the alternative state. Right? So this is sort of plotted by the bar here. So all the orange colored SNPs are ones where Denisovans have an allele that's different from Neanderthals and Africans. And so we only look at the sites where Denisova 2, so our target individual, also has one of these alleles. Right? And so if this is truly heterozygous, then you would expect roughly 50 of the times to have the sites with roughly 50 proportion, and they are still not significantly different from 50%. And outside the regions, in these shaded areas, you can see that we don't find any, or maybe the response, but otherwise we don't find any sites where the two genotypes match this Neanderthal, which is sort of again I think evidence that this is actually a true interest region. We can do a very similar thing with a fragment on the next chromosome of chromosome 8. Here it's even simpler because chromosome 8 is as a male individual, so there's only one chromosome from the heterozygous individual. And so we see that whenever Neanderthals have a particular allele and Denisovans don't in this blue region, with the exception of one step, Denisovans matches Neanderthal. And outside the region, it consistently matches the Denisovan allele. So moving on, we can also ask, this shows right that there has been quite a lot of gene flow early on from Neanderthals into Denisovans. We can also look at the earlier Neanderthals and ask if they retrieve any Denisovan ancestry. And the answer to that is also yes, using this new method, using this Denisova for all Neanderthal data. Again, using the exact same approach, show that this individual has a bit more older, so there's 7 tracts that are between 1 and I think 1.5 centimorgans long. But it has multiple regions with Denisovan ancestry. And the same is true, but to a much lesser extent for this much more younger, recently, I think the publication came out this week, actually in PNAS, this Trieste 8 individual, which is coincided very close by. And there's some, but much less Denisovan ancestry in this individual. In contrast, we don't find actually any Denisovan ancestry in later Neanderthals from Europe, even though this is the region where we have by far the most Neanderthal samples. And both high and low coverage individuals from Europe, mostly late, also earlier Neanderthals don't show any evidence of Denisovan ancestry, which suggests that even though that gene flow might have been quite prevalent in the Altai region, little of that gene flow made it back to Europe. And then you can also look at the high coverage Denisovans, and so this is the tricky analysis because we also use it as the reference. But you can also show that even this late individual seems to have some Neanderthal ancestry here on chromosome 6. Anyway, so as a summary, there seems to be quite a lot of gene flow, and almost for all Denisovans that we have sufficient data to like that there's evidence for gene flow from Neanderthals, right? And also vice versa. For samples from the Altai. And the question is like, what scenarios could lead to such an observation? Right? And so my thought is that this might be some kind of hybrid zone or sink habitat. That the Siberia is not where Neanderthals and Denisovans were at very high prevalence or very high population density. So there were many individuals moving into this region, but comparatively few individuals moving back to maybe the higher density Neanderthal region in Europe, or higher density Denisovan population in some unknown place, like people like to speculate about. And so that's it. So to summarize, there's a new method that I learned to you about inferring ancestry in ancient genomes. I have a preprint, the software is in GitHub, and we're now working on, and so using this criterion for current gene flow between Denisovans and Neanderthals in the Altai region. As an outlook, we also been looking at other scenarios involving modern humans in particular, where there's a lot of ancient DNA data, and a couple of weeks ago, Gendo submitted a pre-printed bioarchive where we look at, using this method to look at Denisovan ancestry in East Asians, where we looked at 2 early East Asian genomes, one which has been published before, that's 40,000 years ago, and Salkit, a new 34,000-year-old Mongolian genome, and where we do show that they also have some Denisovan ancestry, which is not the case for any or almost any of the other early modern humans that we've been looking at. It seems to suggest that Denisovans interacted with the earliest East Asian, or one of the earliest modern human populations that moved into East Asia, and subsequent analysis showed that this ancestry likely remained in the East Asian populations up to the present day people. Yes, that's it. I'm very happy to acknowledge lots of helps with this project from all the ancient Neanderthals in today. Yeah, David, Janet, Svante, and Matthias, and then all the students and postdocs we had the pleasure to work with here. And I hope my sound issues were not too terrible, and there's still someone here.
All right, thank you, Ben. So a really cool method. That's a really cool results. But yeah, unfortunately, there was some major sound issue. But hopefully, on the process YouTube video that will be available afterwards, maybe it will be a bit better. But we will take some questions for the talk. So you can either write here in the Zoom chat or raise your hands in the Zoom chat, or you can write a comment on YouTube stream or on the Twitch channel, and we'll keep track of it all. So please ask questions to Ben.
Seems like I shoot every way away successfully.
Maybe I can start with a question. If you can hear me.
Yeah, sure.
Yeah. So you showed that the method that it works better the longer the diversion time. So at least I think that's what you showed. But then, so was that does it matter when the integration happens? So it's I assume it's better if it's more recent than later.
I mean, essentially what matters is the time between when integration happened and the sample, right? The shorter this time, the easier to detect.
Okay.
Like that.
That means this method can work on a wide range of other problems and species where you have integration that's not too recent, but of course not crazy old either. But how old or which scenarios would it work in?
Yeah, I mean, it's a good question. I think there might be something that one needs to, I mean, in the sort of Denisovan scenario, I think I went to the limit, right? There essentially showed for the sort of most extreme scenario, there is almost no, very little that can be detected. So in the, I mean, there's two things, right? So one is the fragments are very short, and short fragments are harder to detect, particularly using this method. So I mean, practically, I think on the order of like 2,000 generations, one should still have power, but much more than that would be difficult. But I think that's something one need to test based on any particular scenario.
And how hard would it be for somebody else to use the method? Is it all nicely done, so it's just it works straight out of the box, or is there a lot of fiddling around with parameters if we were to use it on a different species?
I've been working with someone that has been trying to do it run it on macaques. It mostly runs. There's some issues in that there's some stages that I need to figure out where, for example, you assume that the individual has 23 chromosomes at most, and there's somewhat silly things like that are not terribly hard to figure out. In general, I try to implement the software in a way that it will be straightforward to run, and at least here other people have run it on their data, which is human data. So at least for humans, I think it should be not too hard to get it to run. For other organisms, probably contact me. And I can see that I fix out all the issues, because most testing has been done on or essentially all testing has been done on either simulations or human data.
Okay, thanks.
There is another question from Mick. It's, have you looked into how high quality the reference source panel needs to be?
Not in great detail. So I assume for simplicity that it's that you have some high quality genomes for the sources. So I don't assume there's any error in the sources for the time being. I think that's something that could be relaxed. It's yeah, it could be relaxed. I'm not quite sure how easy it would be. But yeah, one need to think about exactly how one would add, I guess, further uncertainty to that. But I think in principle it should be possible, but I have not done that yet.
Okay, it looks like, oh yeah, there's another question from Andrea Cabrera. It's, do you need chromosome level reference genomes?
I mean, you not necessarily, I mean, in terms of chromosome level, I assume you don't need full, I mean, you can have, say, if you have a chromosome that, or a species, right, where your chromosome exists as a bunch of scaffolds that you cannot fully resolve, then you can run the analysis just on each scaffold. What the model will not do is try to infer ancestry tracts across different scaffolds, obviously. But you can sort of pretend that each scaffold is its own chromosome, and it will work on that. If that is what you're referring to. But you do need some kind of genetic map.
Okay, it seems that that is all from questions. So thank you very much, Ben. It was really great talk.
Thanks, everyone. And again, I apologize for the audio issues.
Next talk we are having on Monday at 2 p.m. Central European Summertime, and we're going to have Matteo Fumagalli. So please join us and see you at the talk. And thanks again, Ben.
Yes, thanks, Ben.
Yeah, well, my pleasure.